Welcome to our first lecture on bone. We started with our connective tissue, then we moved into our supportive connective tissues, the more specialized ones. We talked about cartilage, now we're moving into bone. Now, bone is either going to form originally from germ cells such as mesodermal mesenchymal cells, or it is going to replace cartilage in some areas. And we'll talk about that in our second lecture. But first, we're going to talk about the structure, the proper structure, if you will, of bone. Now, when we look at the bone matrix, we're going to have organic and inorganic components of the bone, so we need to understand the molecular components that we have. We're going to identify the cells of bone. We're going to have cells that deposit bone, much like we had the cartilage depositing cells, and we're also going to have cells that remodel and reshape bones as they grow. Then when we especially look at long bones, we're going to list and identify the different regions of these bones, the shafts of the bones, the head of the bones, and we're going to give them specialized names. So as far as the structure of bone in this lecture, this is what we hope to accomplish by the end of our podcast. Now, bone is the hardest substance in the body, and I think a lot of times we think of teeth and bone as being synonymous tissues. However, what I want you to realize is that bone is very dynamic. Bone is filled with blood vessels, it's filled with nerves, and it's different from the enamel of teeth, which is non-living tissue. In teeth, we have the blood vessels and nerves underlying the enamel, but in bone, it courses throughout. And because of that dynamic nature, bones can certainly be areas where we can have a reservoir for minerals. As we see, 99% of calcium is stored in bones. And if you didn't realize calcium was important before you took human physiology, hopefully that's one thing you took away from that class, is the understanding of how critical and almost ubiquitous calcium is throughout the body and important for function. Certainly we know our long bones are present for support. They help us for levers that our skeletal muscles are attached to for movement, and as well as protecting our organs. Rib cage is going to be protective. Our cranium is going to be protective as well. Now we begin looking at the cells that are going to give rise to the bone, and we can refer to these initially as osteoprogenitor cells. You might think of them as stem cells in a way. They're going to be mesodermally derived. These mesodermal germ cells are going to give rise to mesenchymal cells. These are pluripotential cells that can become a number of different cell types, such as cartilage, bone, fibroblast. So in this case, these osteoprogenitor cells are going to be those cells derived from the mesenchyme that are going to give rise to bone. And the osteoprogenitor cells, where we find them, we're going to find them on the inner layer of the periosteum. And as we look at our bone, we're going to see, we just like cartilage, we're going to have an outer fibrous and an inner cellular layer of that peripheral tissue. Perichondrium in cartilage, it's periosteum in bone. We're going to describe an organization in our compact bone that are called haversian canal systems, and lining these canals are going to be our osteoprogenitor cells, and then the endosteum on the inside of the bone next to the spongy bone. Again, these are terms we're going to get to as we look at our structure of bone. Now, when we look at bone, we are going to see that these osteoprogenitor cells are going to be very active in cell division, and they're going to give rise to osteoblasts, or they can also give rise to chondrogenic cells. And that's again speaking to this pluripotential nature of those mesenchymal cells, and the special conditions are growth factors and certain chemokines that might stimulate the regrowth of cartilage, especially when we look at how broken bones are repaired. So first we look at osteoblasts. These are going to be the cells that begin depositing bone, and they are somewhat homologous to our chondroblasts. They're going to begin to lay down this bone matrix, and once they're completely surrounded and present inside this lacunar space, then we're going to call them osteocytes. So it's very similar to what we saw with cartilage. Now, osteoblasts are going to produce certainly the organic component of bone, and these are the cartilage fibers, proteoglycans, glycoproteins that are present in the matrix surrounding these cells. But unlike cartilage, this bone matrix, once it becomes calcified, it is going to be impervious to any diffusion of material. So the way these cells are going to enable themselves to be nourished and exchange their waste products are through a series of connections from one cell to another. I like to think of it almost like a bucket brigade. And so these osteoblasts are going to be attached to their neighbors through cell processes, and at the end of these processes are going to have gap junctions. So cells are going to be able to pass along this chain all the way from the periphery of our organized bone unit all the way to a capillary bed. And in that way, this exchange of material through this syncytial network of osteoblasts, they're all going to be nourished, they're all going to be able to give off their waste, and they can survive. Now, while these cells do produce the organic component, when we get to the inorganic component, the calcified nature, this calcium phosphate rich hard material, that is going to be excreted by exocytosis, and we're going to go through specifically how we see that getting accomplished. Now, in this process, we're going to have this organic matrix, and as we're beginning to lay down this calcium phosphate, initially it's not going to be calcified, and so we call it osteoid, and it's going to stain in a different color that we will see with calcified bone. So we have first cartilage, then in some cells we do, and then we're going to begin to remodel and reshape that with the osteoblasts that are then going to begin to lay down this bone matrix as osteoid, and then the final step is the calcification of that osteoid into mature calcified bone. So in this illustration, what we're looking at, we are looking at our osteoblasts, which we can see lining this particular piece of bone. Here we have bone, and you can see how this bone is a light purplish color, and this dark, dense purple is calcified cartilage. Now, this calcified cartilage is the beginning process. Some people refer to it as primary bone. As cartilage becomes calcified, that cartilaginous matrix that's much more dense in the chondroitin sulfate proteoglycans is going to stain that more basophilic color. And that calcified cartilage is going to be replaced, and it's going to be replaced with our secondary bone, and you can see this here. The osteoblasts, you can see on the outside, they are depositing bone matrix, and so this bone will be growing in this direction. We also have osteoblasts over here. Some people will refer to these as bone lining cells. I don't particularly care for that, but you can see how they would come up with that terminology. Now, once these osteoblasts completely surround themselves with bone matrix, and we can see one here, then that is an osteocyte now present inside this lacuna of the bone itself. Now, osteoblasts are going to be responding to their environment. They're going to be responding to external cues as to the extent of their activity. And so on the cell surface of osteoblasts, we have integrins, and remember these are these dimeric transmembrane molecules, alpha and beta subunits, that interact with the extracellular matrix, the collagens. In this case, we're going to have osteonectin as a cell adhesion molecule. But we also have parathyroid receptors present on the osteoblast. Now, these are going to respond to osteoprotegrin ligand, OPGL, and this is going to lead to the development from our osteoprogenitor cells into osteoclasts. We haven't got to these, but these are the cells that are going to remodel and reshape bone. Osteoclast stimulating factor is another factor that is going to lead these progenitor cells down an osteoclastic pathway instead of an osteoblast pathway. Now, osteocytes are the ones that have completely surrounded themselves with the bone matrix. And as I mentioned earlier, the interconnected cytoplasmic processes, these long, thin threads that are connected with gap junctions that allow these cells to communicate with one another, these are referred to as canaliculi. Now, in many instances in your textbook and in diagrams, you're going to see stained spaces, and a lot of times you're going to see the spaces that are left behind in bone where the soft tissue has been removed, and you've got these little tunnels that are still left in our calcium phosphate hard bone matrix. The cells, the processes, the canaliculi are gone. Some people still refer to those spaces as the canaliculi. Those are actually the spaces where the canaliculi resided. So I want you to understand there's a difference. But many people just refer to the spaces as well as the cytoplasmic processes as canaliculi. And that's not exactly correct, but we will stick with that for our class. I just wanted you to know the very specifics of using that terminology. Now, around the osteocyte, there is a periosteoitic space. This is basically the lacuna that surrounds the cell, and you have fluid-filled material that surrounds the osteocyte. It's going to be somewhat of a shock absorber type of fluid that's present in there, and it suspends the osteocyte within the lacuna. That way the osteocyte's not just slamming against this hard matrix of calcium phosphate material. And when you add up all of the fluid that's present in all these periosteoic spaces of all your bones, it's going to be over 1 liter of volume for the average sized person. So that's quite a bit of fluid volume, quite a bit of space. And yet another reason why we must stay hydrated all the time. This is not going to be exchanged as readily as our fluid that's in our circulatory system, but certainly a very important fluid reservoir in our bodies. Here we have a scanning electron micrographic view of 3 osteocytes. Now, you say they're not completely surrounded because we're looking at them. Well, some of this bone has been removed, and what you're looking at are portions of a lacuna and the cells that were present within those lacuna. And you can see the canaliculi extending from one cell to another. There's just many of these processes. And in this case, you can see how they extend to one another. Now, if the matrix were on top of these, you can understand how we would have to have a little tunnel in that calcium phosphate matrix for these processes to extend. So those are the canaliculi. Now, when you look at a light level picture, they're going to look like little fibrous hairs. Now, this is a Haversian system. We're going to have a central Haversian canal. This is where you will find the nerves and the blood vessels, capillaries in particular, and connected from these, you've got your osteoprogenitor cells or your osteoblasts lining these Haversian canals, and these are going to be connected to all the underlying cells through these little hairs that you see, almost like cracks in this matrix. Those are the canaliculi. Here you can see an osteocyte. Here's another osteocyte with its canaliculi, and all of these are going to be interconnected. And we're going to lay down new bone in concentric rings. That's why it appears that you have sort of a stratified layer here. You can see most of these osteocytes within the same plane as we go around and look at this organization. But there you can see the canaliculi extending to and from our osteocytes. Now, we mentioned that some of these osteoprogenitor cells and even osteoblasts, when they receive a signal, they can become osteoclasts. Now, these are the cells that are responsible for remodeling the bone. We refer to it as bone resorption. They actually go in and degrade both the organic and inorganic portions of existing bone. When we look at our legs or other bones in our body that are subjected to different forces, depending upon what forces we subject our body to day after day after day, our body is going to account for that and remodel our bones. When we look at, for instance, archaeologists and paleontologists, when they look at bones of Egyptians that were present in areas surrounding some of the pyramids and the Sphinx, they know that some of these were slaves that did really, really heavy, hard manual labor, because when they look at the bone, they can see it was thicker on one side than the other to support that additional weight and keep the legs from bowing outward. So that's a way that we remodel bone, lay down new bone in ways that's going to be most beneficial to what we face each and every day. Now, osteoclasts are extremely easy to distinguish from the other cells in the bone. They're going to be extremely large, 150 microns in diameter. To give you a frame of reference, a red blood cell is about 8 microns in diameter. But not only that, osteoclasts are going to be multinucleated. Now, we're not talking about like binucleated, which we saw in our transitional epithelium, but we're talking maybe up to 50 nuclei that we have present in here. They are motile. They can move around, but typically you're going to find them existing in a hollowed out depression present in the bone that they have created, and that's called a Hauschup's lacuna. So even though they can move from place to place, typically that's what you're going to see beneath the osteoclast is this Hauschup's lacuna. So in this micrograph, we see a lot of cells that are present. We've got our osteoblasts, or bone lining cells. We have the cell that is completely surrounded by bone. And what do we call that one? It's an osteocyte. And although this osteoclast hasn't really begun to shallow out a depression yet, we do see 1, 2, at least 3 or 4 nuclei present in this particular image. And you can see how much larger it is than the other cells we have present in the field. That is our osteoclast. Now, when we look at the osteoclast, especially on that basal area beneath the osteoclast and just adjacent to the bone, we can identify different regions of that particular cell. So first, we're going to see that we have a basal zone. Now, although we're talking basal zone, I want you to understand that we're really talking about the cell here, the floor of the cell itself. And this is where you're going to find the nuclei and the organelles. But then as we look at the vesicular zone, this is going to be more basal or closer to the bone than the nuclei. So don't let that basal term throw you off. The vesicular zone is just that. We have a ton of vesicles that are going to be secreting materials to help degrade this bone and resorb that material. Now, as we look at the clear zone, this is going to be an area within the cytoplasm that is just directly above where we have the site of bone resorption. And it's a clear zone because we have a thick actin ring, and this ring is going to go around the circumference of the cell, and it's going to create essentially a Loctite surface with the bone. It's almost like that suction cup that you used to play with when we'd shoot the darts. It's the edge of that cup. It's going to sit down on the bone and help create a really watertight seal so that the materials we're secreting directly underneath the cell to degrade that bone and create the house chip cocoon is not going to get out. Now, as we look at what's directly in contact with the bone, we're going to see the ruffled border. This is going to be the most basal of all these areas, and we refer to these ruffled borders being present in what is called the subosteoclastic compartment. So this is going to be immediately between the cell and the bone. We call that the subosteoclastic department, and it's going to extend down into that zone. So as we look at our osteoclast, we see our multiple nuclei in this particular field. We have some mitochondria. Then we have our vesicular zone, which is very easy to see. Then we've got our ruffled border extending down into this subosteoclastic compartment. And this inset is blown up so you can see these finger-like projections. And remember, we're increasing the surface area here, which is both going to be for excretion and absorption of material. And the actin ring, the clear zone, would be present over here. I'm not seeing a real good distinction between the vesicles and the actin ring, but the clear zone is going to be out in this region. Now, how do we resorb bone, this calcium phosphate rich matrix? And what we're going to be using is we are going to be using carbonic anhydrase. Now, carbonic anhydrase, we have heard of this before in physiology in a number of places. Red blood cells in the kidney, well, we're using it again. Because what's going to happen, carbonic anhydrase from the osteoclast, they're going to form carbonic acid. Now, carbonic acid is going to break down into our hydrogen ions and our bicarb, and that's going to form inside the cell. But we can't allow the hydrogen ions to form at too high concentration because that will lower our pH too much. So the hydrogen ions are pumped out rapidly into the subosteoclastic compartment. And when you do that, that is going to decrease the pH, which is going to begin to dissolve those inorganic components of the bone. Now, that's going to be just the calcium phosphate rich matrix. And once that's broken down into its subunits, then that can be reabsorbed in the osteoclast and passed on to the capillaries. So the lower pH takes care of the calcium phosphate, the inorganic part. The osteoclast also produces hydrolases, collagenases, gelatinases, these different hydrolytic enzymes that are going to degrade the organic component of the bone. Now, collagenases, they are going to degrade native collagen. Gelatinases are enzymes that break down denatured collagen. So gelatin is a term for denatured collagen. So you've got two different forms that are working on the same molecule, just in its different conformational states. So here we have a diagram showing our osteoclast. We are combining carbon dioxide with water to form carbonic acid. There we're pumping out the hydrogen ions. But we're also going to be importing our absorbed material that we can then pump to the capillary along with the sodium bicarb. Now remember that bicarb, we're going to balance that out in our kidneys to be able to utilize it or remove more of it as needed. Let's talk about the inorganic part of bone just a little bit. This calcium phosphate rich matrix basically is going to form what we call hydroxyapatite crystals. Now, the dry weight of bone, remember, if you take bone and you desiccate out all that periosteoitic fluid that are around the osteocytes, if you remove the fluid in the Haversian systems, the blood vessels, 65% of the bone is going to be this hydroxyapatite crystal. It's a very dense material. The other is going to come primarily from the cells, from our collagens and our proteins, but most of bone is going to be this hydroxyapatite crystal made of calcium and phosphate. Now let's look at the bone. As we look at the structure of bone, first we're going to look at the two different types of bone. Now we're not talking about long versus flat bones. We'll get to that in a minute. But as we look at this histological description of bone, in long bones, and you might think of this as your femur, for instance, you can see on the periphery of this long bone, it's going to be very, very dense, and we refer to this as the compact bone. Now, once we move to the inside where we have our marrow cavity, we are going to have spongy bone, and this is where we're going to have a lot of our hemopoietic precursor cells producing bone. We're going to have a lot of our white blood cell precursors in here as well. Fat's going to be stored in here quite a bit. And so this is going to be part of the marrow cavity in our spongy bone. So these are the two types, compact on the outside for strength, and then spongy bone, which is going to give us strength but also space where we can accomplish other things such as the development of many of our important cells. In this illustration, we can see once again we've got our compact bone on the outside. Here is our periosteum. So if we're looking at an inset from the middle of a long bone, such as our femur, here we can see the arrangement of our compact bone and then the arrangement of our spongy or cancellous bone. Now this thing that looks like a birthday cake or a wedding cake, what that is a sort of a relief illustration trying to show you these concentric rings that are going to be laid down to form this one unit of bone, which is called the osteon. And we'll be talking about that more as we move through and talk about our compact bone. Now this cancellous or spongy bone, we can define it as a red or yellow marrow, depending on if it's red, which we're forming the blood cells, such as our erythrocytes, or if it's yellow, it's going to be a reservoir for fat. And it's the yellow marrow in particular that dogs and carnivores are really going after when they are chewing and gnawing on bones. They're trying to get to that marrow cavity because it's very, very rich source of energy, and that's what they're trying to get to. Now, as we look at a long bone, you saw in our illustration earlier, the shaft of our long bone, this is referred to as the diaphysis. The end or rounded portion of our long bones is referred to as the epiphysis. So as we look at how bone is going to grow, we are going to have our spongy bone, we're going to have our compact bone on the periphery of the diaphysis, we'll have our marrow cavity in the center. And as we get closer to the epiphysis, we're going to have a region that is known as the epiphyseal plate, also known as a growth plate. Now, bone is not going to grow extremely rapidly once it forms. So the epiphyseal plate is made up of hyaline cartilage. And we're going to see how this hyaline cartilage undergoes very rapid mitosis to form additional cellular layers for the lengthening and the extension of these long bones, especially during puberty. Then once we form that cartilage, that's going to be replaced by our bone that is forming subsequent to that. Now, we have the epiphysis we mentioned and the diaphysis. The region that's sort of in between, this transition region between the diaphysis and the epiphysis, is referred to as the metaphysis. So our epiphyseal plate is just after the metaphysis of the bone. That's more of an anatomical term. So here we see, again, this illustration, the epiphysis. Have two on each end, the diaphysis in the middle. We're going to have our compact bone. We have our spongy bone. Periosteum, connective tissue on the outside. End osteum is going to be the cells lining the very inside of the marrow cavity. And then we will have our epiphyseal plate. But they're not really showing us well in here where the epiphyseal plate would be. So, similar to what we saw in cartilage, we have our periosteum, which is going to be our dense, irregular, collagenous connective tissue. Now remember, the only place we're going to find dense regular are in our ligaments and tendons. So all of this other denser material is going to be irregular. We've got the outer fibrous layer, and then we've got the intercellular layer where we find the osteoblasts and our osteoprogenitor stem cells. So once again, we see our bone, we see our osteocytes present within the cavity, and as we move out toward the periphery, we can see our space, some connective tissue fibers out here. So this portion would be our outer fibrous portion of our periosteum, and this would be the inner cellular portion of our periosteum as we're transitioning from connective tissue directly into bone. Now, when we looked previously at our fibrocartilage, that interconnecting point between the patella and our ligaments, what we are going to see is the way that bone is able to hold on is part of the periosteum and part of the collagen of the bone itself. And if you pull some of the periosteum, in this case, away from the bone, what you're going to see is that you have these perpendicular fibers running from the periosteum into the bone. They're interconnected one with another. These are referred to as Sharpie's fibers. And this is going to hold that periosteum attached to and is a part of the continual collagenous network of the bone itself. So in that way, the periosteum is going to be interwoven with our fibrocartilage, that's interwoven with our ligaments and tendons, well, our ligaments in this case, and then that is the way that we are able to attach this connective tissue to the bone itself. Now, we've talked about the long bones quite extensively, but we also have the flat bones, such as the bones that are present part of the skull, the calvaria. And in this case, what we're going to have is we're going to have our inner and outer layers of compact bone. Think of them almost like an Oreo cookie. And these are going to be the tables of our compact bone with spongy bone in the middle, and these are the diploe. Diploe. And the diploe is where we're going to see our marrow cavity, which has some fat storage, and we may have some hematopoietic cells, but typically those are going to form in the marrow cavity of long bones. Now, in this case, when we look at the periosteum, we're going to see that this is attached to the outer table of our flat bone, and we're looking at the skull here, so some people refer to this as the pericranium. So when you look at the bottom table, that is going to have a periosteum, but it is not going to be a periosteum that we would recognize. It is going to be the dura mater, which is directly the first of the meninges adjacent to our brain. So as you're coming down, we've got pericranium, outer table, diploe, the inner table, and then the dura mater. That's going to be the organization of our flat bones. So here we can see a section through some of our bones, our flat bones. We've got our outer table. We got our pericranium. I believe it's not a great picture. I believe this is the skin of the scalp. Here we have our compact bone. Again, inner table, spongy bone in the middle, Inner table of compact bone. And here we have our dura mater that's facing the brain, which is out of our plane of section. Now, to get a little more detail about the organization of bone, we're going to look first at compact bone. And our compact bone, even though its unit is going to be osteons, those wedding cake-like concentric ring structures, and you think of them like the rings of a tree. In this case, when we're looking at these outer portions of compact bone, we're going to see that they are laid down in sheets or lamellae, almost like wafer-thin sheets. And as we're moving in from our periosteum, we're going to see that we have an outer circumferential lamellae, And this, again, is the outermost region. Sharpie's fibers are going to be attached here. And then adjacent to the marrow cavity, we're going to have an inner circumferential lamellae. So in our illustration, we can see the inner part of our marrow cavity. This is our spongy bone. You can see our osteons beginning to form. We're going to have first an inner circumferential lamellae. Our osteons are going to be existing, and as we move out, we're going to have our outer circumferential lamellae as we get to our periosteum. Here you can see them identified as such: inner circumferential lamellae, outer circumferential lamellae. And there you can see our osteons in cross section looking like the concentric rings of a tree. In a histological diagram, once again, we can see this is going to be fairly easy to identify after we've seen this for some time. We see adipose tissue. We can see a lot of our hematopoietic precursor cells, so we are going to quickly realize once we really get to studying bone that this is our marrow cavity. So this is going to be our inner circumferential lamellae. You can see how it is flowing in this direction. This is a bone in cross section, so of course it's going to be flowing around like a ring. And as we move a little bit deeper, we can begin to see our concentric ring structures, our hollow haversian canal. So this is going to be our compact bone and our haversian systems. So we've got our inner and outer circumferential lamellae. Now the osteons, and again, these are those functional units of bone, the ones that look like the rings of trees, the concentric layers of a wedding cake if you do them in relief. We call them haversian systems because they are surrounding the haversian canal. As I mentioned before, this contains blood vessels and nerves, capillaries present there. And these osteons may be anywhere from 4 to 20 concentric rings in size. Now, the fewer the rings, the younger the osteon. As the osteons grow, they're going to deposit more and more and more rings. The youngest ring will always be the one closest to the haversian canal because our osteoprogenitor cells line that haversian canal. So here we see two osteons. This one has a number of layers and concentric rings, so this is going to be an older osteon. Then we see here fewer numbers of osteons, and I wish they had made that canal a little bit bigger because at first the osteon is going to have a very large diameter haversian canal and it's going to continue to get more smaller and smaller and smaller. I like this particular plastic model in this illustration because it's trying to show you the lamellae that's present out here on the outer circumferential lamellae. Here we have the spongy bone that's lining our marrow cavity. The spongy bone will be on this side. So we're looking at these osteons. Now that's a nice three-dimensional relief. Again, when we look at our typical cross-sectional look at the osteons, they're going to look like that. There you see the haversian canal in the center, and we can see our concentric rings. All of these black spots are going to be our osteocytes. We've got our canaliculi, the fibers extending between, and then lining that haversian canal, it's going to be our osteoblasts, or osteoprogenitor cells. And you can see in some ways these run into each other. They're not going to necessarily be perfectly formed concentric circles in every single area. You can see here's a fairly large one, and this one is kind of overriding it. So we're going to look at that overall structure of these haversian systems as they form our compact bone. Now the haversian canal, this is going to be the canal that's running parallel to the length of the osteon. You can see that right in the middle when you cut the cross section. However, Volkmann's canals are going to be perpendicular. They actually interconnect haversian canal systems. They're going to allow blood vessels to move in and out from those haversian canals. And if I can just go back just a few, here we can see our haversian system with the blood vessels and nerves running parallel to the osteons. These would be our Volkmann's canals that are running perpendicular. And when we see those in cross section, what they are going to look like is what you see right here. You can see where this is in cross section, that's a haversian canal, but now we see an extended canal running perpendicular. That's going to be a section through a Volkmann's canal. And in many instances, you'll see these running away from or toward other haversian systems. Now, the cementing line, when we look at the oldest lamellae, the outermost circumferential lamellae, the periphery of that osteon is going to form the cementing line. That is just the beginning part of that osteon as a unit. And as I've mentioned, the newest concentric layer of the osteon is the one closest to the Haversian canal, where our progenitor cells are, our osteoblasts. So in this case, we can see we have a couple of osteons. The cementing line for this osteon would be in this location. Cementing line would be about in this location. It's sort of difficult to see on these. Cementing line would be about in this location. Now, in addition to the lamellae that represent our Haversian system, the concentric rings, we're also going to have lamellae that are left over after osteoclasts have remodeled bone. Osteoclasts aren't necessarily going to get rid of everything. So in this illustration, we can see that we have an osteon, again, made up of a couple of different layers. Here we have our Haversian canal in the center. We can see how it has overridden this osteon, which means this osteon is older than this one, because osteoclasts have remodeled or taken away this portion of this osteoclast to allow this one to grow. This osteon is younger than this one. Same thing has happened. This osteon is younger than this one. However, this osteon is younger than this one. You can see it's overridden. However, in some instances, you do not see Haversian canal associated with any of the lamellae. And right here in this location, you can see some old lamellae, and there's no Haversian canal system that these would go to. This is what is referred to as the interstitial lamellae, simply left over from what osteoclasts began to remove, but didn't necessarily finish that process.